Think forward. Think Research Channel.
The impact of the biological revolution in genomics is poised to really shake the world in medical practice. We're already beginning to see this in areas of genetic diagnostics, susceptibility to disease, pharmacogenomics, how people respond to drugs, identification of new targets for therapeutic intervention. And one can actually picture the day when you'll have a credit card size thing in your wallet which will have your whole genomic, functional genome on this credit card. And a physician will be able to look at this and determine what you should take, what you shouldn't take, and how you should manage your lifestyle, perhaps. And of course, this has huge implications in ethics and privacy issues, which a number of these will be covered in the talks today, as well as the impact of genetics and future medical practice. And as I mentioned, it really is starting to happen now. This year's symposium is entitled Genomic Medicine. It is sponsored by the Office of Research and Graduate Studies and co-sponsored by the Center for Genetics in Health and Medicine and by Pfizer. I'd like to introduce Mike Benke. He's professor of biostatistics at the School of Public Health, and he's going to chair the first session on population scale studies in human and genetic health.
So it's a real pleasure for me to moderate this session on population scale studies of genetic health. This is a really timely topic, as the last 25 years have seen a revolution in our ability to carry out these sorts of studies. In the last 25 years, we've seen human gene mapping move from linkage studies based on a few blood group and enzyme polymorphisms and candidate gene studies of single gene variants in an ad hoc sampling of genes to linkage studies based on large sets of informative microsatellite and SNP markers, candidate gene studies in which we can assay essentially all common variants in essentially any gene, and now genome-wide association studies of thousands of individuals genotyped for hundreds of thousands of markers. These changes have been made possible by remarkable advances in molecular genetics, statistics, and computer science. An example of this trend is the cost of genotyping. 25 years ago, genotyping was laborious, only moderately accurate, and expensive, with per genotype costs on the order of dollars or tens of dollars. Today, genotyping is still hard work, but it's largely automated, highly accurate, and remarkably inexpensive, with per genotype costs measured in cents or tenths of cents. Our two speakers in this session are amongst the leaders in bringing about and taking advantage of many of these remarkable developments. Our first speaker this morning, and the keynote speaker for this conference, is Dr. Francis Collins. It's a special pleasure for me to introduce Francis, since he and I arrived at the University of Michigan in the same year and have been collaborating ever since. Francis did his training in Virginia, Yale, and North Carolina, and joined the faculty at Michigan in 1984. He stayed at Michigan for about 10 years, developing and refining key techniques in positional cloning and applying these techniques to identify genetic variants responsible for cystic fibrosis and neurofibromatosis, and to play a leading role in the identification of variants responsible for Huntington disease and ataxia-telangiectasia. Francis left Michigan in 1993 to serve as director of the National Center for Human Genome Research, now the National Human Genome Research Institute, and so to be the head of the Public Human Genome Project. In the last 12 years, Francis has brilliantly navigated and overcome the scientific, political, and interpersonal obstacles to complete this amazing task on time and under budget. He's gone on to coordinate the sequencing efforts for other organisms and has led the way towards the annotation of the human genome and the cataloging of the common human genetic variants. Remarkably, while an NIH Institute director and carrying out these projects for the Genome Project, Francis has continued to carry out his own research. He and his colleagues positionally cloned genes for multiple endocrine neoplasia and progeria, and together with my group at Michigan and colleagues in Finland and Los Angeles, identified variants near HNF4 alpha that appear to increase risk to type 2 diabetes. I could go on with a long list of more accomplishments and many honors, but rather than taking any more of his time, let me stop now and introduce Dr. Francis Collins.
And it's great to be here at the University of Michigan, seeing so many old friends and seeing how flourishing this environment is. I, as you heard, spent 9 years here, and I think most of what I learned about how to do research and how to operate a laboratory and how to build a group and find interesting problems and work on them with other people are things that I learned here. And it's always wonderful to come back to this remarkable environment. And it's particularly good to see so many students here today. You all are blessed by a wonderful institution that offers many opportunities, and I'm sure you're absolutely reveling in them, and I hope you'll revel in them today, because this is really quite a nice lineup of presentations that have been organized by the people who put this together, and I know they worked pretty hard on it, because I got a lot of emails about all of this. Miriam, you put a lot of time into this. I can vouch for that, as did the others. So I am going to talk about large-scale genomic enterprises, but I'm going to take the liberty of actually inserting into this a few vignettes of specific applications that come from my own research lab, and some of those are large-scale and some are not. But maybe it will give some kind of a spectrum sense of the things that are possible now that we have all of these tools in genomics. Ray said that a geneticist thinks that systems biology is when you work on 2 genes instead of 1. Well, a genomicist thinks, unless you're working on all of them, you're just not really there at all. So whether or not we live up to that is another question, but that's our dream. Well, as was mentioned, the Genome Project did what it was claiming it could do, and did so in a remarkably short period of time, actually, when you consider all the tasks that were set in front of it. And it was my great privilege from '93 onward to lead the U.S. effort in this regard, and to have a chance to work with just an amazing group of people who tackled these very ambitious milestones one by one, developed technologies that hadn't been invented yet in order to make them happen, built facilities to do high-throughput research that were really quite unique and unprecedented in biology. And during the second half of this, really cranked up the sequencing enterprise to the point of sequencing 1,000 base pairs a second, 7 days a week, 24 hours a day, in order to make it possible to have a draft of the genome by the summer of 2000, published in February 2001. And in April 2003, the essentially complete version of the human genome sequence. But mostly what I want to talk about today is what happens after that, the to be continued, because in many ways the Genome Project was a building of a foundation, and now we have the chance to take that foundation and apply it, and particularly to apply it for medical purposes. And that is what I want to talk to you about. So let me first tell you about some of the resources that are being developed in this next phase that are freely accessible, especially with lots of students here. I hope you know about all these things, but I'm not sure that we always have gotten the word out about many of the aspects of what genomics is doing that provides information either in silico or in actual biological materials that you might find very interesting. And these things are moving very quickly. In the first place, sequencing the human genome was great, but we couldn't really interpret it without comparisons. So we're certainly making maximum use of the experiments of evolution to figure out which parts of the genome have been subjected to evolutionary conservation, assuming that those must be, therefore, functionally important. And the bottom line is it's about 5% of the human genome that shows evidence of conservation. That's not to say the other 95% is not doing anything, but it's certainly to say that the 5% that does show conservation is doing something, and we would be well served to look at it most carefully. And only about 1/3 of that is accounted for by known exons. So a lot of the rest of it is really interesting, but we don't know what it does. The sequencing of other organisms is moving forward at a prodigious pace, with now almost 30 mammalian genomes either in the pipeline or present already as a draft. And the most recent one of these being the chimpanzee, published in Nature just about a month ago, and obviously an organism of great interest because it's being our closest relative. But I think our goal here is no less ambitious than to do a sufficient amount of sequencing of a sufficient number of branches of the mammalian radiation to be able to deduce exactly which base pairs in the genome have been subject to selection. And it is feasible, with a lot more work, to actually get to that point of being able to say for each nucleotide, was that nucleotide under selection or not? And that would be pretty powerful in terms of identifying even small motifs, such as things that bind transcription factors, which are otherwise difficult to see if they're not surrounded by other larger motifs that have also been subjected to conservation. Here's an example of the kind of thing which you can get out of very deep sequencing of multiple species. This comes from Eric Green's work, and Eric is focusing on a subset of the genome, and we don't have sequences of this depth and quality of lots of the rest of the genome, but this is a glimpse of where we're going. This happens to be a small part of the cystic fibrosis gene. That is an exon up there of CFTR. What you're looking at down here are all of these comparisons of genome sequences from the orthologous regions of these organisms, and the height of the green bar tells you just how similar they are to the human sequence, which is being used as the test in this circumstance. And I think you can see when you get up high in this diagram, closer to humans in evolutionary terms, the similarities are so high that it's hard to make much of them. As you move down from primates into mammals, then you can see there's a lot more jagged nature to the comparisons. But it would be difficult, I think, with your eye to pick out exactly what are the things that are most conserved here, except perhaps for this, which all the way down to fugu tells you that that's an exon, and of course that's true, it is. Now what you see in this track here is a conservation score, and this actually comes from former Michigan graduate student Elliot Margulies, who, working with Eric Green, has come up with a number of algorithms for determining, with this kind of sequence data, where are the places that evolution has cared about the most. And those are sometimes described as these MCSs, multi-species conserved sequences, and that tells you where those are. And I hope you can see that in addition to the exon, which we sort of knew about already, there are also these other conserved elements in an intron of CFTR. Now, some of those are still mysterious. Of course, the exon is not. This one, interestingly enough, picked out by evolution, turns out to be an alternatively spliced isoform, which had been detected a few years ago and thought to just be noise. But since evolution has cared about it, it probably isn't noise. This is probably a functional isoform of CFTR that had not previously been appreciated. Over here is an interesting one where you have clearly evidence of conservation by these two methods of analysis. And what you're seeing here are these are also the sites for DNase hypersensitivity sites. Again, highlighting the work of former Michigan students. This is the work of Greg Crawford, who's a postdoc in my lab, who's developed a method to find DNase hypersensitivity sites across the entire genome. And this happens to be where two of them fall in this CFTR region. So there's probably something pretty important going on there in terms of regulatory signals. Notice there's still some unexplained things here, so that each one of those could potentially become a research project. And this is not a particularly densely packed area as far as conservation. You'll see this almost wherever you look. And it certainly tells you that if you want to take the stance that introns of large genes like CFTR are just filler, that must not be true. Evolution would not have done all of this work if that were the case. So this kind of conservation opens up a new window into what parts of the genome are functional, although it doesn't tell you what they do, only that they must be important. In that regard, a project which Rick Myers is going to tell you more about is one that I think would be worth your paying attention to, especially because all of its data is up on the web, and particularly at the UC Santa Cruz site. The ENCODE project, Encyclopedia of DNA Elements, is made up now of more than 24 research groups that have all gotten together to work on the same 1% of the genome, a carefully chosen 30 megabases, and apply a whole host of different approaches to try to understand function of that 1%. And again, Rick will tell you more about this. This is great fun watching how this all comes together with these various approaches being compared and contrasted. Another resource I wanted to mention, in case any of you are laboring without knowing about this and could save yourself a lot of time, is an effort to get into the public domain full-length cDNA clones of all the human and mouse genes. All of us who've been around for a little while will recall those times of laboring mightily over many months trying to get the last 5' exon of your favorite gene and having a very hard time getting the cDNA fit together. Well, that shouldn't be necessary. We ought to just do this in an organized fashion, and that's what the MGC, the Mammalian Gene Collection, aims to do. We're now at about the 2/3 point for human and not quite that far for mouse, but quickly getting there in terms of having those full-length cDNAs that are available in high-quality form. They've been carefully sequenced to very high nucleotide certainty, and you can order those from any one of a number of different distributors. So all you have to do is go to the site, see if your clone is there, order it up, and save yourself all of that trouble. This is also extending to rat and to xenopus and to zebrafish. And we do aim in the near future to take these clones and actually flip them over into expression-ready vectors in order to make them even more useful for people who want to do protein expression. Another major effort that is just getting underway, an RFA having been issued about two or 3 weeks ago, is an effort to knock out all of the mouse genes. Obviously, a place like Michigan, where a huge amount of fabulous mouse work has been done, you all will not need to be reminded how valuable such mouse models are for understanding biology and human disease. But I think the time has come where doing knockouts, even though it has been an interesting and educational experience, it's time to move it into a high throughput environment. NIH is spending a vast amount of money on doing these in a one-off fashion. And in fact, we could probably do them about 10-fold more efficiently if we just did this in a fashion where it was done in one place with a particularly high throughput available technology. So there will be an effort now to, at least as ES cells, knock out all of the mouse genes that have not already been knocked out and are not already available as null alleles. And that turns out to be only a few thousand out of the roughly 20,000. So this is going to be an effort over the next 4 to 5 years. Just in probably tomorrow, there will be an announcement also that NIH, by the good graces of multiple institutes getting together, has acquired several hundred mouse knockouts that have already been generated in the private sector, and we will now move them into the public domain, out into the sunshine. And these are extensively phenotyped mouse knockouts that have been generated primarily for the pharmaceutical industry. So it is going to be a lot of things like GPCRs that you might be interested in looking at. And that announcement will be coming out in the next day or so, and those mice will then be freely available without any restriction on use. And I think that's a nice hors d'oeuvre for what is going to be a longer-term effort to get all of the mouse genes knocked out. Another resource just getting underway, and which is a new approach for many people working in genetics, is the availability now in academia of the opportunity to screen very large, complicated libraries of small molecules to see if you can find compounds that act as agonists or antagonists for your favorite protein target or pathway. The centerpiece of this is a series of 10 screening centers, and they've now been funded. And they basically will allow an investigator who has come up with an interesting assay to submit that assay to the screening center, have it run through a library of 100,000 or more compounds, and then you get the compound back to see how you might want to use it. Most of these will be useful primarily as research probes. A few of them may be interesting enough that they'll be picked up by the private sector and carried along into potential clinical trials. There will be a database already up and operational for the last year that collects all this information so you can compute on it. And this is a way of empowering academic investigators who generally have not had access to this approach to add it into their whole armamentarium of how you tackle a difficult biological problem. Again, this is available to investigators who want to apply for using these centers. You simply have to describe your assay, explain how it can be adapted to a high throughput environment, make it clear why you're doing it. There's a peer review step. If you do well in that, then you get into the queue for one of these centers. And all of this can be read about at the NIH Roadmap website, because this is one of the roadmap initiatives put together by Elias Zerhouni. Another major project that I think has profound implications for our understanding of human genetics and applications to medicine is the HapMap project. And I'm going to outline a bit for you the motivation for this. And I know Aravinda Chakravarti will be also saying some things about this project, one that he's been deeply involved in as a co-chair of the analysis group of this enterprise. This is just like, in some ways, the sequencing effort. This is an international project involving 1,000 or more people working across country lines to try to generate a public database of information about the genome. This time, it's about the variable part of the genome as opposed to the 99.9% that we all share. The motivation for this is a medical one. Although we are learning things from this about human population history, the motivation here is really to find the genes that cause common diseases, like the ones you see here, which I think anybody who's worked on this would agree is really hard. Mike and I will vouch for that, having worked on diabetes now, type 2, for more than 10 years. This is really hard. And it's hard because of the power issues. The approach that most of us have taken in the past is to use linkage analysis, which works great for Mendelian disorders, but which is not all that powerful when it comes to common disorders where you know there are multiple gene variants involved and no single one of which contributes a very large quantitative amount to the disease risk. And Risch and Marikangas documented mathematically why that was almost 10 years ago. And they argued that if you really wanted to increase your power, you'd be better off to be able to do an association study comparing cases and controls rather than family-based linkage for a common susceptibility variant in a common disease. The problem has been that even though that was true, association studies until recently were limited to basically candidate genes. And if you accept the fact you wouldn't be doing this anyway if you already knew the answer, then the candidate gene list that anybody would put together was usually not very accurate. And oftentimes we ended up with nothing, or you ended up with a weak positive which didn't replicate in somebody else's study. So we were kind of in a dilemma. Linkage was underpowered, association was limited to candidate genes, and we weren't smart enough to know which ones to try. I think that's all about to change. Let me motivate for you how this might work. Because actually, compared to linkage analysis, which is fairly counterintuitive to many people, association analysis is very straightforward once you get the hang of it. I know we have a diversity of folks here, so let me just walk through how this would work. So here's some sequence from chromosome 7, about 2,000 base pairs. There are 3 places in this sequence where there are common single nucleotide polymorphisms, or SNPs, marked with the yellow arrow, where people will differ in that particular nucleotide. So if you wanted, for instance, to go out and find the variants in the genome that are associated with type 2 diabetes, well, one of them might be one of these 3 you're looking at, but there's about another 10 million out there that you'd want to look at too. So a straightforward way, intuitively satisfying way to try to find the variants associated with the disease would be to simply test every one of them. So this would be a whole genome association approach. First, you would need to identify those 10 million common SNPs. Then, because you're looking for subtle contributions, small numbers of cases are not going to serve you well. You need a big study. So let's say 1,000 cases and 1,000 controls. That's the right order of magnitude. Obviously, more is better, but there are limits to what you can afford to do. And actually, the cost here is primarily going to be the clinical work of collecting and phenotyping all of these individuals, because you've got to do that and you've got to do it right. And then you just simply have to genotype all the DNAs for all the SNPs. Well, Mike mentioned that genotyping costs are coming down, so maybe this isn't so bad, but this is 20 billion genotypes. So even at a penny a genotype, that would be $200 million for each disease. If you, for your October 1st deadline, send in a grant to the NIH, with this kind of a budget, I suspect you'll get a quick call back from a very distraught review officer saying, I'm sorry, we can't accept this because, you know, things are tough around here. NIH is not rolling in money this year. So this is theoretically satisfying, but practically impossible. And maybe we'll get there as the genotyping costs come down further, but we're not there today. Well, maybe that was overkill. And this is what I want to motivate for you, is that HapMap basically allows you a shortcut, so you don't have to pay those $200 million, because, in fact, it was overkill. You've got 3 SNPs here. Do you really have to check all 3 of them? Or if you checked one of them and didn't find anything, would that tell you that you can walk away from this particular part of chromosome 7, saying there's nothing here of interest? The real question then is, are the SNPs correlated with their neighbors? Do they travel together in neighborhoods? Well, in fact, they could occur in 8 different haplotypes. There are 3 SNPs. Each of them has 2 possibilities, so that's 2 times 2 times 2 is 8 haplotypes diagrammed here in a simple way where I'm leaving out all the nucleotides that are constant and just showing the ones that are variable. But if you do the experiment for this part of chromosome 7 on hundreds of people, you will find, at least to a first approximation, 90 to 95% of the time, only 2 of these are observed. And that's interesting, and it tells you something about our history as humans, that we haven't been around that long, and there hasn't been time for scrambling of these haplotypes by crossing over, although it now appears that crossing over is not something that happens randomly anyway. It happens in hotspots. But what this says in a practical sense is, if you were looking in this region trying to figure out whether there's a diabetes susceptibility gene here, you could have tested any one of these three and gotten the same result if you tested the other two. So it's not necessary to look at all of them. One will do. The question, of course, in terms of how useful this is what is the distance over which this kind of relationship, which we call linkage disequilibrium, between adjacent SNPs applies. And on the average, although there are wild differences in this, it's in the neighborhood of 20 kilobases. A little less than that in African populations, maybe a little more in European and Asian populations, and that's all very consistent with what we know about bottlenecks and population histories. But that's not bad, because in a 20 kilobase interval, you'd expect to have 60 or 70 SNPs, and you might need to only check 2 or 3 of them in order to decide whether that interval contains something interesting or not. So the HapMap Consortium, who you see here, the leaders of it, gathered at a meeting less than a year ago in Japan, has been laboring very intensively over the course of the last 3 years to try to generate the map of how this linkage disequilibrium relationship exists across the genome. I said that on the average it extends over 20 kilobases, but there's some segments that are much larger than that and some that are much smaller. And at the moment, we do not have a reliable way to just look at the sequence and figure out the boundaries of that LD relationship. So you have to do it experimentally. And that's what this group has been doing, using a set of 270 DNA samples that are collected from different parts of the world. Now, first of all, we had to be sure we had enough SNPs to work with. If you look at the curve of SNPs in the database dbSNP, it was at about 2 million when HapMap started, primarily because of the SNPs consortium. And then we turned our sequencing machines toward this problem, now having really prodigious sequencing capacity in the public project, and we're able to build up that number now to well over 10 million SNPs. If you're looking for a variant in the human genome that has an allele frequency of 20% or greater for the minor allele, it is very likely that it's already been found and it's sitting there waiting for you in dbSNP. Here's a little bit of data. This is to show you what you find in the DNA samples that have been looked at by HapMap. These are 2 different regions of the genome. This is chromosome 2. This is part of chromosome 7. The distance across from left to right is about half a megabase, about 500 kilobases. And these funny-looking diagrams basically show you the degree to which SNPs are traveling together, the linkage disequilibrium. So when, for instance, you see a triangle like that, that says that the SNPs in that interval in these DNA samples are very tightly associated with each other. And so that's a pretty big region, almost 100 kilobases, where the SNPs are all traveling rather in lockstep, and where you would not need to check more than a few of them in order to decide whether there's something interesting there. Now, these 3 different diagrams are the 3 different samples that were used. At the top, these are DNA samples from the Yoruba in Ibadan, Nigeria. These samples are from members of the Seth pedigrees in Utah, so these are Northern European in background. And this is a sample which is half Han Chinese from Beijing and the other half of the samples from Japanese living in Tokyo. And I hope what you can see is that the similarities are actually quite impressive in terms of the degree of linkage disequilibrium. There is somewhat less in Africa than there is in the other 2 populations, but the patterns are in fact fairly similar across the groups. There's more data here I don't have time to go into, but this is the kind of information that's coming out now across the entire genome as a result of HapMap. Again, HapMap does not make an effort to try to collect any information about clinical phenotypes in the individuals whose DNA is sampled. So this is not going to tell you which of these are associated with any disease, but it gives you a research tool to go out and find those answers, and that's what we now hope will happen in a big way. So HapMap milestones. February 2005, phase 1 map was completed with over 1 million SNPs genotyped. Those have been extensively analyzed, and there's a manuscript that will be coming out in the not too distant future describing what's been learned from that. But because genotyping costs came down so quickly, we were able to mount a phase 2 that hadn't really been planned initially, but we had the time and the money left to do this. So Perligen has added another 3 million SNPs to the map. That data has been deposited in the HapMap Consortium's database. And in fact, just yesterday, 6 chromosomes worth of that were, after they've been cleaned and appropriately represented, were put up on the HapMap website. And now we are going to see the analysis of that combined data set of phase 1 and phase 2 coming up during the fall. And of course, the big question for many people is, okay, how did it work? How much of a shortcut is this really going to be? How many SNPs do you now have to test if you want to look across the whole genome? And again, depending on which algorithm you use, you get a slightly different answer, but something in the neighborhood of 250,000 SNPs will give you 80 to 90% coverage of the genome for European or Asian samples, and perhaps 400,000 for African samples. So we're talking about a savings here in terms of the amount of work you've got to do, somewhere in the neighborhood of 30 to 40-fold, which is pretty nice. And if you're sort of interested, you can also pass your time by playing HapMapopoly, which is a little bit of amusing Monopoly-like game put together by the folks at Cold Spring Harbor to pass the time. So now how about this? How are we going to use this? Well, again, you've got this resource. I'm going to come to whole genome association in 1 second, but first let's point out that if you are sure you're interested in candidate genes and you really do know which ones you want to test, this gives you a chance to test them really in a rigorous way. A lot of candidate gene analyses in the past have come up empty, but maybe actually didn't look broadly enough at regulatory variants, for instance. With this approach, you can effectively say there is or there is not a common variant in a particular gene region that's associated with disease. Secondly, you can take a linkage peak that you may have arrived at after considerable pain and suffering, but it's a big, broad peak for a common disease, and you're sort of stuck there. Now you can actually pepper that interval with HapMap-based SNPs and see whether you can identify an association under that SNP. As Mike mentioned, he and I have been working together over the course of quite a while, about 10 years, on the problem of type 2 diabetes in a collaboration that involves both Michigan and Bethesda, but also our good friends in Finland who are doing all of the subject recruitment, and our friends at USC who have been involved in the physiology of this. And several of Mike's group are here today at this presentation. Just a very quick vignette. The way that we initially approached this was by doing linkage analysis on affected SIB pairs, because that's sort of what we had available to us beginning many years back. And the result of that gave you a bunch of LOD scores, with the most impressive one being on chromosome 20, and several other groups also found evidence of linkage to that part of the long arm of 20. But it's a big, broad, jagged, nasty-looking peak, and it was really hard to figure out how you would go from there to an actual causative variant. As HapMap began to mature and the density of SNPs got better and better, we were able then to look at this interval by an association approach. And out of this came this result, which seems actually quite logical in retrospect, but at the time was a big surprise, which is that, in fact, there are variants in the neighborhood of the alternative promoter of this gene called HNF4 alpha. And you can see association of those variants with type 2 diabetes. Now, this is a candidate gene that probably would jump out of the list at you anyway, because mutations in the coding region of HNF4 alpha cause maturity onset diabetes of the young, MODY, the disease that Stephen Fyans here at University of Michigan defined many years ago. And in fact, somewhat to our embarrassment, we had previously published that this was not the gene responsible for this particular linkage signal, because we'd looked down here and we hadn't found anything in the coding region. As you can see, life can be more complicated than that. Another example about how using a HapMap-based approach allows you to be more rigorous about electing or not electing a particular candidate. We found those variants, colleagues at St. Louis, Alan Permit, were able to identify the same SNPs showing association in an Ashkenazi set of cases and controls with type 2 diabetes. And multiple other groups have now tested this, and most of them have in fact found the same association with the same SNPs or SNPs nearby. Now notice all of these are showing association because they're all traveling together. I'll come back to that in a minute. The good news is,
Think forward. Think Research Channel.